Great. Well, thank you, Amanda, and welcome everyone. It's really great to see you all here today. As Amanda mentioned, we just concluded our scientific sessions last week, and I'm happy to report that there were a number of really important findings and what I would call really critical insights that will shape and inform ongoing and future research. So, and that's really the value of these annual conferences, is just bringing everyone together that's working on RUNX1 and having the opportunity to share. We had over 180 different scientists join us across the 3 different days. And so, and I would say, even though it was a virtual format, we had some really rich discussions. So as Amanda said, Dr. Speck and I will attempt to summarize what we think were the key takeaways for all of you in about 15 minutes. And then we'll introduce you to 2 physician scientists who are relatively new to our community. You may, if you were at the meeting last year, you may have met them. But they wanted to say hello and tell you a little bit about the work that they're doing and how you might consider participating so that you can be a part of further accelerating research. So, and then at the very end, we have about 30 minutes. At the end, we'll have time for a Q&A session. So before we jump into the key takeaways, it's important that I remind all of you about some of the cell types we're going to talk about and the names we'll be using, so that you can stay with us as we try to explain some of these key takeaways. It can help to use analogies, and so we'll use analogies. The analogy Dr. Speck is going to present is a little bit different from what we've used in the past, but hopefully looking at things with different analogies will help solidify some of these concepts even more so. What I wanted to make sure that you're all crystal clear on is the specialized cells that exist in every single tissue in our body. So like the blood, the skin, the brain, there are these cells called stem cells. And these cells are special because they can divide to become different kinds of cells within that respective tissue. So in the bone marrow is what we talk about here a lot with RUNX1 FPD. That's really the source of our entire blood system. And that's where the stem cells, or the blood stem cells, reside. So if you watch some of the educational videos that Amanda sent out at the end of last week as a primer, in those videos, I used the green lawn as an analogy of the blood system, and green seeds as the stem cells that produce that green lawn. And so the stem cells in your bone marrow are responsible for generating all the different blood cell types, including platelets, red blood cells, white blood cells, etc. And in fact, before you're even born, while you're in the womb, is when all of your stem cells are generated. And this is similar to the eggs in a female ovary. A little girl is born with all of her eggs for the rest of her life. Well, that's the same. The same is true with blood stem cells. And each of these different stem cells we call a clone. And they're essentially identical, which is why they're termed a clone. But we have specialized tools now in the lab that allows us to track these different stem cell clones and how they're behaving. And so one really key point here is, why do we care about stem cells? Why are we talking about clones? And that's because a blood cancer is actually originated or generated from a stem cell clone, a clone that went bad, that turned into a bad clone. Usually we can identify it because it has new mutations or cancer-causing mutations. So going back to the lawn analogy, it's a green stem cell that produces the green lawn that turned into a weed. And now you've got weed growing out into your lawn, and that essentially becomes your cancer. Is a blood cancer. So hopefully that gives you some important terminology as we talk about some of the key takeaways. All right, so with that, I'm going to turn it over to Dr. Speck, who is the chair of our scientific advisory board, and she'll talk about the first few key takeaways. So let me advance the slides here.
Hi, everyone. It's really great to talk to you again. I look forward to next year when I can actually meet with you again, because that's one of the most rewarding aspects of this meeting for me. So I'm going to talk about some key takeaways. Some of them are key new questions that we hadn't really considered before because we didn't have enough information to realize that these were questions. So one of the questions or key takeaways was about the continued uncertainty about B core mutations. Are they good or are they bad or both? So if you advance to the next slide. So here, my analogy here is that every single one of these schematics of a person actually represents an individual hematopoietic stem cell. And so when you're young, you have this large cohort of very healthy hematopoietic stem cells. So all of those will perform well. And so, but over time, due to stress, Katrin, if you move on to the next one, including the stress of aging, our stem cells begin to decline. And this is a natural process. It occurs in everyone. So some of them are just plain exhausted and can't do their job very well anymore. Some will actually senesce or die. And one of the things that we think is happening is the decline is likely happening faster in RUNX1 FPD patients. So if we go to the next slide, the first step in the development of a leukemia is that one of these hematopoietic stem cells acquires a mutation in a gene. And B core mutations are very, very common first mutations in RUNX1 FPD patients. So they're found early, even before there's any evidence of malignancies. And what we've come to learn based on some investigators' work is that HSCs and RUNX1 FPD seem to really like getting B core mutations because if you go to the next slide, Katrin, some actually acquire several independent B core mutations. So just because they're very common though, and often the first to occur, this doesn't necessarily mean that B core mutations will contribute to the leukemia. They may not be helping the leukemia. It could be that they're just rescuing these stressed HSCs and making them stronger and perform better. So work from Ravi Majeta's lab actually showed that B core mutations helped RUNX1 mutant HSCs perform better. So they may actually be a good thing. This sort of raised the question of whether B core mutations are kind of a good thing because they're rescuing the stress, rescuing stressed RUNX1 mutant HSCs. But if you go to the next slide, the other possibility is that B core mutations could actually generate very aggressive HSCs that create problems, and these eventually evolve into leukemia cells. And we really just don't know yet which way it is. So it may, but if they're really kind of good mutations that allow HSCs to perform better, we may not need to eliminate them. But if they're aggressive if they create aggressive HSCs, we probably will want to be thinking about eliminating them. So fortunately, we have multiple animal models now to help us figure this out. So for example, Ravi Majeti's lab has this human mouse model. So what he does is he makes B core mutations in isolated human hematopoietic stem cells, and then he transplants them into mice in order to study them. Serene Avigan has a zebrafish model, and my lab has a mouse model of B core mutations. And each model has its strengths and its weaknesses, so they complement each other, and they'll help us figure out what's going on with these B core mutations, whether they're good or bad or both. Next slide. So another key takeaway was that the rise and fall of these hematopoietic or stem cell clones over time illustrates the dynamic and actually unpredictable nature of stem cells in the bone marrow. So next slide. So this slide sort of represents what can happen when a hematopoietic stem cell acquires, let's say, a B core mutation. On the y-axis, what I've graphed is the percentage of blood cells that have have actually have this B core mutation. And you can see that in patient A, the percentage of these B core mutant HSCs as a percentage of the total hematopoietic stem cells, they actually kind of grow over time. So that clone increases in size. However, we're finding from multiple studies that these clones can also be stable for many years. So they just sort of go up and down and up and down, and they go on like that for a while. But in patient C, it shows even clones can disappear. So you can find a patient with a clone, and over time that clone just goes away. And that seems to be particularly true of very small clones. And I think you'll hear more about this from Alex Speck a little bit later during the day. So we don't know why this is a case. We don't understand which way a particular clone will behave. And this is another thing that we need to sort out. Next slide. So another key takeaway is that it's clear that RUNX1 FPD patients have elevated levels of inflammation, including in their bone marrow. And it's thought that it's known actually that inflammation promotes the development of leukemia. So the RRP is funding several investigators who are investigating the potential link between inflammation and leukemia. Lucio Castilla presented some very exciting work at the meeting. So he's testing the ability of anti-inflammatory drugs to prevent leukemia in a mouse model. And he found that blocking one inflammatory molecule or cytokine called the tumor necrosis factor with a drug called etanercept will delay the onset of cancer in his mouse model. There are many inflammatory cytokines that are elevated in the bone marrow of RUNX1 FPD patients. So we were all really surprised that blocking just one of them would have such a dramatic effect. Etanercept is used to treat various forms of psoriasis and arthritis. It has some fairly significant side effects at the doses that patients with autoimmune disease take them, but we're hoping that perhaps lower doses could be used to dampen the much milder inflammation in RUNX1 FPD patients. Next slide. So another key takeaway are that new tools are being developed to help with clinical diagnosis. So a major problem in FPD is RUNX1 FPD is what we call variants of uncertain significance. So somebody comes to the clinic with symptoms suggesting they have RUNX1 FPD, their DNA is sequenced, and a deviation in the DNA of the RUNX1 gene is found. But the results can be equivocal because perhaps nobody has ever seen that change in the DNA before in other patients, or knows whether that particular change in the DNA would cause a problem, is a pathogenic change. And so a diagnosis of RUNX1 FPD can't be made in those cases. So Tim Ripperger's lab has been developing ways, trying to find the most reliable ways to determine whether a mutation actually is pathogenic. And they presented information showing that some assays are very good at detecting this. They are very reliable at predicting whether a mutation will be pathogenic or not. On the other hand, they found that some assays aren't so good for that those same assays aren't so good for some other types of mutation. But anyway, they're using the assays that they're really confident of to analyze new potential mutations that are brought to their attention so that a diagnosis can be made. And finally, for the part of this I'm presenting, a key takeaway is that another tool that's being developed are methods to analyze platelets in RUNX1 FPD patients. So patients' platelets, as you know, these little pieces of cells that are needed for blood to clot, and the activity and the performance of platelets appear to be very good indicators of RUNX1 activity. They perform poorly when RUNX1 is low, and they perform well when RUNX1 levels are just right. And a couple labs are using platelet parameters as another way to test whether mutations could be pathogenic based on what the platelets are, look like and how they're behaving. And also to monitor how well drugs that we're trying in the clinic are being able to improve RUNX1 activity. The thought being that if a drug is improving RUNX1 activity, the platelets will also improve. All right, take it away, Katrin.
Yeah, thank you. Thank you, Nancy. Okay. So I think many of you are, I hope many of you will be excited about this new takeaway. So we have two new clinical trials that were discussed at the conference last week that are coming online or hopefully launching in the next 6 to 12 months for RUNX1 FPD. The first listed there is low-dose sirolimus, or the drug is also called rapamycin. And the rationale or the data that supported the development of this protocol came from Dr. Anupriya Agarwal. And in her studies, where she collaborated very closely with the NIH team, and so thank you to all of you who participated in the NIH study, because the samples that the NIH team were able to collect, they were then able to share fresh bone marrow and samples with Dr. Agarwal at OHSU. And through Dr. Agarwal's studies, she was able to identify that in the bone marrow milieu, which means just the floating liquid surrounding those clones, those stem cells in the bone marrow, there were very high levels of pro-inflammatory cytokines. So these are proteins that generate an inflammatory response in the bone marrow. And when Dr. Agarwal did very careful analyses, she used many different kinds of experiments to try to hone in on, well, which of these cytokines are maybe connected to a internal cell signaling or messaging system where all those cytokines may be channeling through one messaging system within the stem cell compartment. She was trying to identify, is there a commonality that could be druggable? And when she did those experiments, she identified what we term in the science field a signaling pathway, or a signaling pathway, and it's called mTOR. And there happens to be a FDA-approved drug called sirolimus or rapamycin. Those are used interchangeably. And this drug you can use to dampen mTOR signaling. And so now there is a clinical trial in development with using very low doses of this drug, which was originally actually approved for transplant patients who have experienced graft versus host disease. So, but in that setting, the drug is used at very high doses. So in this clinical trial, a very low dose will be used to see if we can dampen the inflammatory response that we think is happening in the bone marrow of RUNX1 patients. So it'll be a pilot study with 6 patients, and we'll see if there's any compelling data to suggest that we're actually doing something positive and reducing inflammation in the bone marrow. Then there's another clinical trial, which you'll get to hear about actually in one of the upcoming sessions by Dr. Tim Olson at CHOP, which is what we're calling a stem cell harvesting and banking study. So that'll allow patients to go in to the clinic and have their blood stem cells pulled out of their bone marrow. Not all of them, of course, because we do need blood stem cells to create the blood system, but at least a portion of those stem cells pulled out and banked. And the rationale for that is to allow for individuals to have access to a future gene therapy study should that ever become available. And we'll get into a little bit more about the rationale and why this is important to do during that session. So hopefully I'm not leaving you with too many questions at the moment, but I do want to make sure that we stay on track with time. So the key takeaway here is that the research community is working really quickly, and it's pretty impressive how quickly we've gone from doing basic science work and translational science into already testing things in the clinic. You'll also hear about one more clinical trial that, if you've been around with us long enough, you heard last year a discussion about a drug called imatinib, and Dr. Leah Cunningham and Dr. Alan Cantor is going to be with us, I think already in the next session or in a couple sessions, to talk about that clinical trial as well. So that means we have 3 clinical trials coming for RUNX1 patients, and it's pretty exciting. The other key takeaway was, what I like to do at these meetings is poll the scientific community and ask, the experts in the field to help us think about where we should be funding. Now, of course, we have a wonderful scientific advisory board, and I'm talking with them, it seems like weekly sometimes. But we also have a very broad, rich, diverse scientific community. And so at the meeting last week, we had several surveys and we had panel discussions where we really asked the community, where do you think RRP should be funding in the coming years? And the answer was, there was really high consensus that at this point, given the information and the knowledge that we have around RUNX1 FPD, is that we really should continue funding a diversified portfolio, which means we shouldn't just fund clinical research, or we shouldn't just fund basic science, but we need to continue to fund through all of the components of the scientific discovery pipeline. So we need to continue to fund basic research so that we can get at answers to questions like, are B-core mutations or clones good, bad, or both? Because that will inform future treatments. But at the same time, we need to place bets, and we need to fund research like the research that Nancy had highlighted, Dr. Lucio Castillo, where he said, hey, I want to try testing TNF inhibitor in a mouse model and see if it works. And lo and behold, we're seeing some really compelling positive data that maybe targeting TNF could actually delay blood cancer, which is incredibly exciting. And had we not said, let's give it a try, we wouldn't have that information today. And then at the same time, we want to support any kind of clinical trial that makes sense as soon as we can. So diversified approach, we will continue. And it's really wonderful when you have that consensus from a whole community that everyone agrees. That makes the decision-making quite easy. And then finally, I wanted to end with what I thought was really encouraging data from a group of European investigators who collected over 160 patient data. They went around to different institutions within Europe and asked, have you ever followed and managed a RUNX1 FPD patient in your institution? And so we had, I think, over 16 institutions participate. And through that study, they identified 161 patients. And what they did was they then looked at their medical records. And within that cohort of 161 patients, they identified 36 patients who did develop AML as a result of their RUNX1 FPD. And then they asked the questions, well, what happened to those 36 patients who developed AML? And what was encouraging was that up until last week, we had all assumed that having a RUNX1 FPD AML is not a good prognosis, meaning it is a very tough AML to treat. And we thought that the outcomes of patients with AML who have an underlying RUNX1 mutation may not be as positive as those who have sporadic AML, so people who don't have an underlying RUNX1 mutation. But the data that the European investigators presented suggested otherwise, that having an underlying RUNX1 mutation doesn't mean that you can't do really well on the therapies that are available today, should one develop AML. So patients were responding well to therapy and doing fairly well in terms of their response to transplant. So this we felt was encouraging to see, and we'll dig into that data more in the future. All right, we went quite a bit over, but I'm going to end there and introduce two of our, as I mentioned, relatively new physician scientists in the community who wanted to just introduce themselves and share a little bit about the work they're doing. So I'll start with Dr. Jimena Jordan Bruno. Jimena?
Hi, everybody. Yes, it's really a pleasure and an honor to be here. I agree with Dr. Speck. It will be great to meet in person, hopefully next year. But last year for me, it was very enlightening to be in the meeting, not only from all the scientific personalities that were there, but also being able to be close to patients and their families and knowing how long it took for them to be diagnosed in the first place. I'm mostly a clinician, but I'm very interested in germline predisposition syndromes. I actually open a clinic where I see patients with RUNX1 mutations, but other mutations as well, and try to do a comprehensive evaluation. And it's more like most of patients comes with their family members, and we try to, address any needs they may have. My project is to have a biorepository that the objective it is to collect samples of patients and carriers of RUNX1 germline mutations in the samples with blood, bone marrow, and maybe skin samples at some point. And the goal is to be able to share these samples with the wonderful scientific community who is doing already, groundbreaking research, is in order to, contribute also to what the NIH is already doing. And whoever is interested in contribute with samples at some point or have a question about this, please email me or my mentor, Dr. Manti Carroll from Penn.
Great. Thank you so much, Jimena. Really appreciate you being here and for all the work that you've and all your efforts in the last year since being such a huge collaborator with us. I'm going to now introduce Dr. Alex Speck from Vanderbilt University, who wanted to just share a few things about the work that he's doing and some opportunities. Alex?
Thank you so much. So again, it was really an incredible experience last year in person in Princeton. And one of the things that we heard from the Research Guided by Patients Committee and the larger patient community was that there's a lot of interest in how patients with RUNX1 have inflammation, not just in their bone marrow and in their blood, but actually how inflammation affects other diseases, such as psoriasis, inflammation in the skin, or inflammatory bowel disease, and inflammation in the bowels. And we took this concept to another very large supporter of biomedical research called the Chan Zuckerberg Initiative, which is supported by Mark Zuckerberg of Facebook fame and his wife, Priscilla Chan, who's a pediatrician. And they were really excited to hear that we're interested in inflammation, looking more broadly. We've worked a lot with colleagues that are supported by this initiative, studying things all over the world. And one of the amazing advances in the past year is that we're now able to take what was basically a trash and turn it into treasure. And I'll tell you what I mean by that. Anytime patients go to hospitals for procedures, it might be a colonoscopy, it might be a skin biopsy. Those biopsies that are used to help diagnose skin disease, colon polyps, are actually stored for about 10 years. Many institutions like at Vanderbilt effectively store them forever because it's too much trouble to go back through and actually get rid of them. And with new technologies that have been developed by investigators supported by Chan Zuckerberg, we have the ability to go back and actually profile these sort of things that are otherwise not being used from biopsies. And so one of the requests that we're seeking to partner with the community on is if patients have had biopsies for any reason, existing clinical biopsies, we'd like for the hospitals that are currently holding them and not doing anything with them to release them for research purposes. And so there's been, I believe, a survey sent around, and we'll engage more with that in the year ahead. But we hope to be able to really learn more about inflammation in places other than just the bone marrow. So stay tuned.
Thanks so much, Alex. Appreciate it. So if you haven't seen the survey, perhaps we'll send out another survey, Katrin. Would love your help on that. And then we'll share more information about the work that Dr. Jordan Bruno is doing. And if you're not already participating in the natural history study and you want to stay with your provider, there are ways for you to share samples. There will be ways for you to share samples through the work that Dr. Jordan Bruno is doing. So more to come there.